And I'm from the Department of Molecular Pharmacology of the University of Groningen. Today's date is March 27th, and this is a recorded class for the course of Pathology and Immunology for the first year pharmacy student. This particular class is going to be about T helper responses and especially the T helper 1 responses. In former classes I talked about how CD4 positive T cells are called T helper cells because they help other cells with immune responses, so B cells and CD8 positive T cells, but they can also help for instance macrophages or eosinophils or other cells to have the best immune response possible, and we'll talk about this part in two classes. This one is about Th1 responses, and there's another one about Th2 and Th17 responses. So what help do CD4 positive T cells provide? Well, we can be exposed to many types of dangers. We can be exposed to in- and extracellular bacteria, to viruses, fungi, parasites, even objects like nails that can be hammered into your skull. So you need a host of different types of help because every bug or thing pictured on this slide needs a slightly different response. So in order to have help for all these different bugs, we have different types of T helper cells. So when a CD4 positive T cell recognizes an antigen in the context of MHC class 2 presented by a dendritic cell or an antigen presenting cell, the antigen presenting cell will tell the T lymphocyte what it needs. This cell gets information from the tissue tells the dendritic cell, hey, I'm infected with a parasite or with a fungus or whatever. And signals from the dendritic cells are then transferred to this T cell, and this T cell knows what the tissue needs. So it can become a Th1, a Th2, or a Th17 cell. Now you might wonder, why is this numbering so weird? Why is it not Th1, Th2, Th3? What happened to 3 through to 16? Well, when lymphocytes were first discovered, we thought there was one type of cell, and then it was discovered that some cells made interferon gamma and some cells made IL-4, IL-5 and IL-13. So when this was discovered, they said, oh, okay, let's call this one Th1 and this one Th2. But then they discovered there was also a cell that made IL-17. And so they decided to name this Th17 to link it to the production of IL-17. So there are actually not any numbers in between, except for one cell, which is called Th9, which makes IL-9. So we're stuck with Th1, Th2, Th17. Th1 makes interferon gamma. Th2 makes IL-4, IL-5 and IL-13, and Th17 makes IL-17 and IL-22. And they all target different cells. So this Th1 cell targets macrophages, the Th2 cell targets eosinophils and mast cells, and the Th17 cell targets neutrophils. So in this class I'll be talking specifically about the Th1 cell. And the Th1 cell helps out macrophages that have phagocytosed bacteria and helps them with killing these pathogens. Sometimes it also has a role in things that are not related to the pathogen. For instance, in autoimmune responses or in chronic inflammation, this cell also produces pathology in those diseases. So here again, in a more conceptual context, here's the lymph node. Here's the dendritic cell coming in from a distant tissue with messages from that tissue. It's presenting an antigen to the lymphocyte in this lymph node, and they can decide to become either Th1, Th2 or Th17, or the CD4 positive T cell can help the B cell to produce antibodies. So these different types of T helper cells can then move out of the lymph node into the blood, into the tissue, where there's help needed with the elimination of microbes. So what is the effector function of a CD4 positive Th1 cell? Well, its most important cytokine is interferon gamma, and it starts producing that when it's being told by a antigen presenting cells that this is the type of lymphocyte that is needed. Then it starts dividing and proliferating and developing into a Th1 cell that produces interferon gamma. And this interferon gamma can stimulate phagocytosis and degradation of microbes by macrophages. And for instance, it is very effective in stimulating the production of oxygen radicals. So this cytokine induces classical activation of macrophages and makes them into mean and lean killing machines. So it means that the microbes that it's phagocytosed can be killed. And it can also help the B cells producing antibodies that help with phagocytosis. So it actually tells the B cells to start producing IgG antibodies. So what is an example of a Th1 reaction? For instance, the reaction to tuberculosis bacteria. Those bacteria are being phagocytosed by macrophages, and these macrophages need help with clearing the infection. So tuberculosis is caused by a bacterium called Mycobacterium, and this Mycobacterium, which is shown here, is a bacterium that you can inhale. So tuberculosis is a lung disease. It also has manifestations outside of the lung, but the primary infection is in the lung. And that's because the Mycobacterium has carbohydrates that are specific for bacteria that are being recognized by a receptor on the alveolar macrophage, the mannose receptor, for instance, and that leads to phagocytosis of Mycobacterium. So the Mycobacterium then gets into a phagosome, and normally the phagosome would fuse with a lysosome to start digesting whatever has been phagocytosed. Well, Mycobacterium has all these ways of preventing that. It will prevent acidification of the phagolysosome. It also can prevent the fusion of the phagosome with the lysosome, and therefore that leads to maturation arrest. So it means that the bacterium cannot be killed effectively, and the bacterium then has the possibility to start dividing, and eventually this cell will burst because there are too many bacteria inside. And then it can spread to other parts of the lung and other parts of the body. So how can a T cell help with this? The T cell that has developed because of the dendritic cell that told it, look, we need Th1 help in the lungs. This T cell then starts producing interferon gamma, and the interferon gamma will activate the macrophage. So the phagolysosome maturation is much better, the activation is better. This activated macrophage will become an M1 macrophage with nitric oxide production of reactive oxygen species that will lead to mycobacterial killing. If it's still not effective enough, if it still cannot kill all the bacteria, then it will try to wall off the infection. It will make what we call a granuloma. So all the infected cells are here, and they're being walled off by other cells that will prevent the spread of the bacterium throughout the body. So more close up, it looks like this. So here we have the infected macrophages that become necrotic and start to fall apart. And here we have lots of mycobacterium species. And the the macrophages in the neighborhood will start to wall off this infection. So they'll go around it and start to become epithelial-like cells, epithelial-like macrophages. And some of the macrophages will fuse with each other, so that's seen here, and they're then called giant cells. So the nuclei of the different cells will not fuse, so they will still be separate, but the cytoplasm will fuse. So you'll get these humongous big cells with multiple nuclei called giant cells. And these help in trying to control the infection. Another example of these giant cells can be found in bone, where the osteoclasts are fused macrophages with multiple nuclei that will help with the breakdown of bone. So here the giant cells help with the breakdown of the bacteria. So around this walled off infection are B and T cells that try to help the macrophages control the infection. So what does that look like in the lung? This is a tissue section. Here you see alveoli, normal lung tissue, and here you see a giant granuloma with in here the necrotic inner part with the bacteria, and this wall here sort of looks like layers is the macrophages that are epithelial-like, and around it you see all these tiny small cells that are B and T cells. Here a close-up of the giant cell. So this is a couple of fused macrophages with all the separate nuclei still within this giant cell. And here's another one, slightly smaller, and you could actually count how many macrophages fused to become this cell. You can also see that they're not all the same. So here's a giant cell that has many nuclei, but they're all huddled together. Here all the nuclei are on the outside. So there are different types of giant cells that help different functions and help in different ways with controlling the infection. So what is a TB infection? What can it be like? Well, there are several ways that the infection can go. You have the primary infection here. That will lead to a localization in the lung where the immune system tries to wall off the infection. If that all is successful, then you get a healed lesion with scar tissue. But if that is not possible, the lesion will be walled off and you still have bacteria that can spread in theory. We call that a latent lesion. But what can also happen is that the immune system cannot control the infection at all, and you get progressive primary tuberculosis. So it will spread in the lung and it will spread to other organs, the liver and the spleen, and this form is called military TB. This form can also happen when you have a latent lesion and for whatever reason you get reactivation of the disease. It will spread, you get localized destructive lesions in the lung or in other organs where it can spread, where the tissue is destroyed by the immune system trying to control the infection. And this can also lead to military TB. So how do you know you've been exposed to TB? For that we do a Mantoux test, and which actually uses the immune system to see if you have been exposed. So if you have been exposed, you'll have had an adaptive immune response. You will have had Th1 cells develop to help the macrophages, and we make use of that in the Mantoux test. So what is happening in the Mantoux test is that parts of the bacterium, from the cell wall of the bacterium, are injected into the skin and that will, if you have been exposed, you will have memory cells, and if these memory cells are reactivated, then you'll get Th1 cells coming into that area where the antigen is, and these Th1 cells will start producing interferon gamma. Then macrophages in the skin will become activated, and you'll get a localized inflammatory response. So if at the place where you've been injected, you'll get a welt with inflammation, that means that you have been exposed to the bacterium in the past. It doesn't mean that you have active disease. That's something you can't tell from this test, but it shows that you've been exposed to the bacterium and you've had an immune response against the bacterium. Here again, what happens to the cells? So here you get intradermal injection of the tuberculin molecules. Here's the memory Th1 cell that will become activated and start producing interferon gamma. The macrophage will be activated and you'll get a reaction in your skin. So that's about Th1 responses. We also have Th2 and Th17 that I'll talk about in the next class.